and then high pressure versus low pressure. And we used a 2x2 factorial design. That means that you got randomized to either receive saline and high, saline and low, soap and high, soap and low. And it kind of looks like this. You get randomized to high pressure, randomized to cast iron, normal saline, etc. And it looks like this in the analysis. So you can analyze the saline groups, you can analyze the pressure groups separately. What is the benefit to this sort of thing, arguably? Well, the benefit is, yeah, 2 trials for the price of 1. You can use the same sample to look at 2 different treatments. What's the problem?
You need more patients.
Well, yeah, there's an issue of patience. And there's, what's that?
The number. You need a bigger trial because you have different subgroups.
That's right. There is patient issues for sure in terms of number, in terms of teasing out the difference between the 2 groups. And the other issue is, well, what about if there's interaction between the treatments? What happens if soap's effect is so much better with low pressure that you start to see different effects and interaction? So there's issues of interaction between the treatments. But we chose to use this design. This was the general makeup of the trial. There was a steering committee of which there's a number of different people, including trauma surgeons, including epidemiologists, including statisticians. There's an, what happened here? I am, but, okay. Anyway, doesn't matter. So there's the steering committee here. We had an independent adjudication committee that was blinded to outcome effects. So there was about 4 of us on that. There's data safety and monitoring. There's a methods coordinating center, and there were participating clinical centers, funneling patients up to the methods center to try and do this. The research questions are here. Impact we already talked about. Secondary, and we addressed the secondary outcomes of functional outcome, delayed union, non-union functional outcome, and all that sort of stuff. But we've, so this is what we've got. 102 patients. We had 9 participating sites. We had 95% follow-up for a year, and we had great compliance rates with the protocol that was developed by the steering committee. Now, I just want to talk to you a little bit about the results quickly. So, we addressed issues of, well, can we work together? Yes. Can people adhere to a protocol? Yes. Can people get follow-up? Yes. And, you know, can we recruit patients? Yes. So, and you had a bunch of charts looking at that. Now, to look at the results, we analyzed them again from soap and saline, and we analyzed them to low and high pressure. Now, let me just talk a little bit about how we're going to present these results. So, here's one way. Well, and these are the real results. And we're just finishing, we're just submitting this paper. So there's no significant difference between normal saline and soap on reoperation rates, and there's no significant difference low pressure or high pressure on reoperation rates. Reoperation rates for infection, non-union, and wound healing problems. Well, what do people think about that? What does it tell us? Nothing. Well, it tells us there's no difference between groups. I mean, there's no difference. So we found no difference. Yeah?
The numbers are too small.
So the numbers are too small. So what does that bring us? Maybe this is an issue of beta error. So we can't say that there's no difference based on this. So this is, I'm just using this as an illustration of one way to present results. Would you want to move forward with a trial of 3,000 patients and $2 million based on this?
Was there a trend at all? Or?
Ah, excellent point. So this is another way to present the results. You know what? There was a trend for a 23% relative risk reduction in risk of reoperation for infection, wound healing problems, and non-union with soap over saline. And there was a trend of a 44% relative risk reduction in reoperation with the use of low pressure versus high pressure. Now what do people think?
Brad?
Yeah.
Well, we were talking earlier about fluctuations in the endpoints, so I don't think that means anything.
Okay, point number 1, doesn't mean anything. Should we go to a large trial or not? You're going to fund it. Yeah.
Something's either significant or is not. A trend could, like 1 or 2, 500 more patients could spin you in either direction. So I agree. It doesn't.
Yep, no, you're exactly right. I'm not. So here's another way to present the results. This is sort of the forest plot way, which because I just like it so much visually, it makes sense. Here's the line of no effect. So here's our point estimate of effect of 23% relative risk reduction, and here's our point estimate of effect of 44% relative risk reduction for soap and for low pressure, and here's our confidence limits. So this, to me, says, well, you know what? We could actually be really helping people, or we could actually be delaying things and causing reoperations. Similarly here. And so this, to me, says, if we're going to do a really large trial, this is compelling to me to say, well, you know what? We're seeing a trend of 23% risk reduction, which we know that surgeons are going to think is clinically important based on our survey. We know that. And 40% reduction in risk is also clinically important based on what people told us. What we don't know is where specifically that effect is going to lie. And we can see that with increasing our numbers, as you've all suggested, to 3,000 people or 4,000 people, the precision is going to go like this, and we may come up with something that's both statistically and clinically significant. Now, this is just one point I'd like to bring out. We have looked at reoperation rates. And it's a composite of infection, reoperation for infection, reoperation for non-union, and reoperation for wound healing problems. What do people think about this issue of using a composite endpoint? Are there any problems? I'll tell you some of the rationale. Rationale for using composite endpoints is if the event rates are actually really low, we kind of need to bump up our event rates a little bit to try and look at something, and so we'll make it a composite. But what do we need the composite to be? We need the composite to be pretty similarly patient important. If we look at, so for instance, and this is in the user's guides. This is another chapter in the user's guides of composite endpoints. If we look at a composite endpoint of death and rehospitalization, and if one or two of these occurs, that's our event rate. What's the main issue here? Well, for me, death is slightly more important than rehospitalization. And so if the main driver of effect is death, I'm going to think one thing. If the main driver of effect is rehospitalization, I'm going to think another thing. So composite endpoints arguably should be similarly patient important. And this is from the user's guides. Questions to ask when you're interpreting composite endpoints. Are the component endpoints of similar importance? Did the endpoints occur with similar frequencies? And do the component endpoints share similar relative risk reductions? Things to keep in the back of your mind when you're dealing and thinking about composite endpoints. Again, here, I would argue that a reoperation for infection and a reoperation are fairly similar patient-important outcome events, arguably. We can argue the merits of infection versus a wound healing problem or infection versus a non-union, but basically you're still taking them back to the operating room. So this brings us to sort of the pivotal trial. And I'll be almost finished. Funding went up. Boom. U.S. Department of Defense gave some money, OTC-AOD gave some money, and the pilot study and this data with a grant application to CIHR gave some money. So now we're dealing with more money, significantly more dollars. One of the funding agencies suggested that, in fact, hey, you know what? We've got guys overseas. These instruments, these guns are quite expensive for us. We have to ship. We want you to add another arm. And the other arm is going to be a gravity flow arm. So we want high pressure, low pressure, gravity flow. Take it back to the steering committee. What do we think? Yes, 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 yes. We're going to bring in another arm, etc. The problems with introducing another arm are issues of sample size and getting the patients and all that sort of stuff. So what can you do to combat that? Get more centers involved. Get more people involved. You've shown an infrastructure already. Let's get more people involved. And so people are actually recruiting in all of these areas across the globe for this particular trial, which we estimate is going to have 2,000, 2,400, something like that, patients. Again, this is the sort of breakdown of the arms. And these are our estimate sample size calculation for each cell in the group. And we're going to analyze that way again, a 3x2 factorial design. Now, I'm going to take you for a detailed look at our protocol. So everybody get ready. Just kidding. I'm not going to do that, obviously. These are some of the slides from some. Yeah, is there questions?
Yeah, sorry to interrupt you, Brad.
No, please.
Like, you just showed that map of the world and all those places you're doing this study in. For something like an open fraction, where you have different types of operating rooms, different types of bacteria, different levels of sterility, I would say, in the operating room. You're not worried about the heterogeneity of this study when you're taking a good, arguably second or third world country.
And an excellent point, and a point that's argued at each investigator meeting that we're at, and by steering committee people and all that sort of stuff. The one issue is, well, maybe you're going to contaminate your samples based on different people's OR experiences. The other issue is, well, you know what? This is a potentially cheap solution that maybe have an effect worldwide. Maybe we want to see what happens in real life. So this is an issue of, efficacy versus our, effectiveness, our generalizability of the trial. And so people argue back and forth. If we include everybody, it's going to make it a bit more generalizable for the world. This is potentially a real cheap solution. Now,
It's not like those two places are a spectrum of problems, they're dichotomous.
correct.
And it only works in Hamilton.
That's right. So one of the ways to combat that issue, it's argued, ethical, unethical, we're using more private hospitals in India as opposed to public hospitals, number one. Number two, if a site has issues with data collection or anything like that, and we're not getting it, then the site is going to be out. Number two. And the other thing was we're stratifying randomization based on site. So we're going to be able to sort of do things like that in the analysis. Is there going to be site-to-site differences? Matt?
On the same train of thought, what about the patient fractures in terms of fracture, open fracture type? Maybe I missed that.
No, you didn't, because I didn't say it. We're stratifying for, which I was going to sort of get to in the protocol, but we're stratifying for open fracture type. So type 1s and 2s, we think clinically is a different entity. Type 3s are a different entity, so we're stratifying for that. So we're stratifying for site, stratifying for open fracture type.
The time is
Yep, sorry?
time is open fracture.
Yep, all caught within the data points. So timing of doing surgery, yes? Yeah, that's all caught within the case report forms. We know that data. We have a data safety and monitoring committee as well that's going to be independently, they're independent of the study and they will be independently looking at the data. I'm not going to go through this in detail, just to say, because it is lunchtime, we have study objectives, inclusion criteria, strict exclusion criteria. We've defined the irrigating solutions. We've supplied the irrigating solutions. We've sort of protocolized what we think is the amount of fluid that you need. We've just defined the pressures. We have people helping us with supplying the guns, strikers supplying guns, so we can sort of get the issues with different guns sort of sorted. We're using 2 specific guns. We have a randomization method, and we have an area of central randomization. I'm only bringing these up because these are going to be concepts that we're going to talk about later on. This is a randomization website. This is what you do. You click next, and you click randomize, and there it is. I have no way of knowing where it is. We have no way of knowing what's coming up next, and everybody does it through the randomization center. We've standardized procedures. We've standardized follow-up. We have primary study endpoints, secondary endpoints. We've thought about adverse events, unanticipated problems, early withdrawal, etc., etc., etc. We've thought about the subgroups. What's going to be clinically important? Well, we think type of fracture is going to be clinically important. So we want to see, is that subgroup any different? What else might be clinically important? Well, we know hands act a little bit differently than tibias. That might be upper versus lower extremity. Maybe that's something different. So we built in some subgroup analyses with this. And the implications are that it potentially could be a cheap solution to open fracture management, arguably applicable, and establish a timeline. So during this last half hour or so, I hope we've just sort of illustrated going from experimental models to finding out what people do, to getting a pilot study together, to going to a full pivotal trial. And we're enrolling for this trial for the past 2 months, I think. So, that's it.